Hi everyone, welcome to NEET Biology Channel. Auxin is used by gardeners to prepare weed-free lawns. But no damage is caused to grass as auxin. A. Promotes abscission of mature leaves only. B. Does not affect mature monocotyledonous plants. C. Can help in cell division in grasses to produce growth. D. Promotes apical dominance. Correct answer. B. Does not affect mature monocotyledonous plants. Explanation: auxin-based herbicides, like 2,4-D, are synthetic and selectively affect broad-leaved dicot plants by disrupting their growth processes. However, monocot grasses remain largely unaffected due to their different vascular arrangement and cellular responses. Therefore, when used in lawns, these herbicides eliminate dicot weeds without harming monocot grasses. Other options either do not explain selectivity, like apical dominance, or are unrelated, example cell division or leaf abscission. Lecithin, a small molecular weight organic compound found in living tissues, is an example of A. Phospholipids. B. Glycerides. C. Carbohydrates. D. Amino acids. Correct answer is A. Phospholipids. Explanation: lecithin is a key component of cell membranes and is classified as a phospholipid. It consists of glycerol, two fatty acids, a phosphate group, and choline. Lecithin is amphipathic and contributes to membrane fluidity. It is not a glyceride, which lacks a phosphate group, carbohydrate, or amino acid. Match list 1 with list 2. List 1 list 2 times A. Xylem fiber 1. Conduct water. Times B. Companion cells 2. Living with sieve tubes. Times C. Sieve tube elements 3. Dead with thick walls. Times D. Vessels 4. Lack nucleus but have cytoplasm. A. A1 B2, C3, D4. B. A3, B4, C1 D2. C. A2, B3, C4, D1. D. A3, B2, C4, D1. Correct answer is D. A3, B2, C4, D1. Explanation: A. Xylem fiber 3, dead with thick walls, non-living, lignified, provide support. B. Companion cells 2, living with sieve tubes, living, assist sieve tubes in transport. C. Sieve tube elements 4, lack nucleus but have cytoplasm, enucleate but living, transport sugars. D. Vessels 1, conduct water, dead, lignified, specialized for water conduction. Why other options are wrong? Times option A, wrong, as sieve tubes are not dead with thick walls. Times option B, wrong, companion cells don't lack a nucleus, that's sieve tubes. Times option C, wrong, xylem fibers aren't living with sieve tubes, they are dead support cells. Thus, the correct answer is option D, A3, B2, C4, D1. Identify the set of correct statements. A. The flowers of Vallisneria are colorful and produce nectar. B. The flowers of water lily are not pollinated by water. C. In most water-pollinated species, the pollen grains are protected from wetting. D. Pollen grains of some hydrophytes are long and ribbon-like. E. In some hydrophytes, the pollen grains are carried passively inside water. A, B and C only. B, A, C and E only. C, B, C, D and E only. D, A, B, D and E only. Correct answer is C, B, C, D and E only. Explanation. A is false. Vallisneria has small, non-showy flowers with no nectar. B is true. Water lily is insect-pollinated, not water-pollinated. C is true. To survive water pollination, pollen grains are hydrophobic. D is true. Examples like Zostera have ribbon-like pollen for water transport. E is true. Some hydrophytes rely on passive water current for pollen dispersal. The list of endangered species was released by A. WWF, B. FOAM, C. IUCN, D. GEAC. Correct answer is CIUCN. Explanation. The International Union for Conservation of Nature, IUCN, publishes the Red Data Book, which contains lists of threatened and endangered species. WWF works on conservation but doesn't issue these lists. Form is irrelevant here, and GIAC deals with GMOs, not species endangerment. What is the fate of a piece of DNA carrying only the gene of interest, which is transferred into an alien organism? A. The piece of DNA would be able to multiply itself independently in the progeny cells of the organism. B. It may get integrated into the genome of the recipient. C. It may multiply and be inherited along with the host DNA. Or D. The alien piece of DNA is not an integral part of the chromosome. E. It shows the ability to replicate. A, B and C only. B and C only. C A, D and E only. D A, B, C and E. Correct answer is B and C only. Explanation. If a piece of DNA is inserted into a host without a vector, it generally requires integration into the genome to be inherited and expressed. If integrated, it will multiply and pass to progeny. Without proper origin of replication, ORI, it cannot replicate independently, thus eliminating A and E. Which of the following are required for the dark reaction of photosynthesis? A. Light, B. Chlorophyll, C. CO, D. ATP, E. NADF. A, B, and C only. B, C, D, and E only. C, A, C, and D only. D, B, C, and E only. Correct answer is B, C, D, and E only. Explanation. The dark reaction, Calvin cycle, does not require light directly or chlorophyll. It uses CO, ATP, and NADF produced during the light reaction to synthesize glucose. Hence, A and B are not needed directly in the dark phase. The type of conservation in which the threatened species are taken out from their natural habitat and placed in a special setting where they can be protected and given special care is called A. Ex situ conservation, B. In situ conservation, C. Sustainable development, D. Biodiversity conservation. Correct answer is a ex situ conservation. Ex situ conservation of species outside their natural habitat. Examples: zoos, animals, botanical gardens, plants, seed banks, cryopreservation, tissue culture techniques. In situ conservation of species in their natural habitats, example national parks, wildlife sanctuaries, biosphere reserves. Sustainable development, using resources in such a way that present needs are met without compromising the needs of future generations. Biodiversity conservation, a broad term that includes both in situ and ex situ approaches. Thus, the most precise answer for the given definition is ex situ conservation. Statement 1. Bt toxins are insect group specific and coded by the gene CRY-IAC. Statement 2. Bt toxin exists as inactive protoxin in B. thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into its active form due to the acidic pH of the insect gut. A. Both Statement 1 and Statement 2 are true, and Statement 2 is the correct explanation of Statement 1. B. Both Statement 1 and Statement 2 are true, but Statement 2 is not the correct explanation of Statement 1. C. Statement 1 is false, but Statement 2 is true. D. Statement 1 is true, but Statement 2 is false. Correct answer is C. Statement 1 is false but Statement 2 is true. Explanation. Statement 1 is incorrect. The gene CRY-IAC is specific to lepidopterans, not all insects. Bt toxins are generally group specific, but not all use CRY-IAC. Statement 2 is correct. Bt produces protoxins that are activated in the alkaline gut of insects, not acidic as the statement incorrectly says. Update. Actually, Statement 2 is also wrong because Bt protoxin is activated in an alkaline, not acidic, environment. Thus, correct answer is A. Both Statement 1 and Statement 2 are false. A transcription unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream ends. A. Structural gene, transposons, operator gene. B. Inducer, repressor, structural gene. C. Promoter, structural gene, terminator. D. Repressor, operator gene, structural gene. Correct answer is C. Promoter, structural gene, terminator. Explanation. A transcription unit consists of a promoter, start signal, the structural gene, coding sequence, and a terminator, stop signal. This entire sequence guides transcription by RNA polymerase. Transposons, repressors, and inducers are regulatory, but not essential components of a transcription unit. Hint 2 always cuts DNA molecules at a particular point called recognition sequence, and it consists of A. 6 base pair, B. 4 base pair, C. 10 base pair, D. 8 base pair. Correct answer is A. 6 base pair. Explanation. Hint 2 was the first restriction enzyme discovered to cut DNA at a specific palindromic 6 base pair sequence. Recognition sequences are specific to each restriction enzyme. For Hint 2, it recognizes a 6 base pair sequence and cuts at that precise location. Which of the following is an example of an actinomorphic flower? A. Cassia, B. Pisum, C. Sesbania, D. Datura. Correct answer is D. Datura. Actinomorphic flowers are radially symmetrical, meaning they can be divided into equal halves along any plane. Datura, thorn apple, has a radially symmetrical flower. Cassia, Pisum, and Sesbania are examples of zygomorphic, bilaterally symmetrical flowers.